Hello, BISC130. This is the beginning of recorded lecture 2.4, where we're going to start and finish chapter 8 on photosynthesis. So, like the last chapter and recorded lecture, cellular respiration, photosynthesis is another big, long, complicated metabolic pathway. But before we get into this, there's some terminology that I want to introduce. So, organisms as a whole can be heterotrophs and or autotrophs. These are both defined in the key terms, but let's look at this visually as I read the definitions. The key terms definition of an autotroph is an organism that gets carbon from inorganic sources. Plants are classically autotrophs. They are getting the carbon that they need to make their molecules from the air. Remember, CO2, carbon dioxide, is not an organic molecule. All these things that do photosynthesis, again, most notably plants, but there are other things that do photosynthesis as well, get their carbon from CO2, get their carbon from the air. In contrast, the key terms definition of heterotroph is an organism that consumes organic substances or other organisms for carbon. So yeah, animals, they're eating stuff, either other animals or plants. They're getting their carbon by eating stuff. Fungi are breaking down dead organic matter, but yeah, they're eating stuff. They're not getting their carbon from the air, certainly. And yeah, microscopic organisms, bacteria, they're also getting their carbon not from the air. So yeah, these are two important terms for this and other lectures throughout the quarter. Heterotrophs and autotrophs. It depends on where you get your carbon. From CO2, from the air, or from eaten stuff. Within these two, there is also something called photoautotrophs. And anytime you see photo, you should think of light. So this is a pretty easy one to deduce. Photoautotrophs are, to read from the key terms, an organism capable of producing its own organic compounds from inorganic carbon and energy from sunlight. So photoautotrophs are just autotrophs that use sunlight for energy. They're getting their carbon from inorganic sources, from CO2, and they're getting energy from light. And yeah, this includes plants, obviously, but I want to point out, it's not just plants. There's some protists. We'll talk more about protists in BISC132 if you take that class with me, but these are single-celled organisms. There are some protists and some bacteria that are photoautotrophs as well. Okay, again, just some terminology here. Now, how about the overview? So we did this for cellular respiration. Let's do an overview for photosynthesis as well. So starting materials, ending materials, not balancing this equation. Photosynthesis is going to start with carbon dioxide. Again, this is about getting carbon from the air. So we're going to take this inorganic CO2. We're also going to start with water. We all know plants need water, and this is why they're actually destroying H2O as a part of photosynthesis. So they're consuming and burning through water in this chemical process. Another starting material is light, obviously, which is going to be a form of energy. So we're going to use these materials and this energy to create C6H12O6. Again, I'm not a fan of making you memorize a bunch of chemical formulas, but we already introduced this one in the last chapter, and it's a pretty important one, glucose C6H12O6. And as a byproduct, we're going to create O2, oxygen gas, as a part of photosynthesis. Now, if you're staring at this and comparing this to the previous chapter, it should actually look a little similar. Photosynthesis is essentially cellular respiration, but in reverse. They're kind of two sides of the same coin. If you remember, cellular respiration took glucose and oxygen and created carbon dioxide, water, and not light, but ATP, created energy. This really is the reverse of that process, building something instead of breaking something down. Now, I should point out that for eukaryotes, or virtually all steps of photosynthesis, I'll point out the one that's not, virtually all steps are taking place in the chloroplast. Remember these specialized organelles for doing photosynthesis, and in eukaryotic photoautotrophs, almost everything happens in these organelles. Now, just like we broke down cellular respiration into a couple of parts to make it a little simpler, we could break photosynthesis down into a few parts as well. We could essentially divide this in two. We can split photosynthesis into the half that needs light and the half that doesn't need light. So the first half is called the light-dependent reactions, or, because who doesn't like a nice acronym, the LDR. These are the reactions that are going to actively require the presence of light in order to happen. The other half is sometimes called the dark reactions, but that's misleading, I think, because it implies that it has to be nighttime. The other half, a better name, is light-independent reactions, or LIR. It doesn't need to be nighttime. It could be daytime. These reactions simply do not require light. So it could be day, it could be night, whenever the LIR does not require light to happen. So obviously, we're going to start with the LDR, but before we get into the light-dependent reactions, I think we need to talk a little bit more about light itself. So light, what we call light or visible light, is a type of electromagnetic radiation. So yeah, you may recognize this from a physics class. We got gamma rays, X-rays, microwaves, radio waves. All these are the same thing. All these different things are really the same phenomenon, and gamma rays and microwaves and radio waves and red light and green light, all these things are electromagnetic radiation. The stuff that we call visible light that we're able to see with our eyes is just a very specific type of electromagnetic radiation that has a very specific wavelength. The only difference between gamma rays, which would kill you pretty instantaneously, and radio waves that are passing harmlessly through all of us right now, and green light, which we're all kind of seeing on the screen right here. The only difference between these things is the wavelength of these different waves. So in summary, visible light is a type of electromagnetic radiation. Visible light has a specific wavelength, which we will not be memorizing, but yeah, there are numbers associated with this. Even more specifically, if we want to talk about our different colors of light also have different wavelengths. Again, the only difference between blue and green and yellow and red is the wavelength of that light. And here's another figure kind of showing these waves. Waves tight together, waves more spaced apart. That's what I mean by wavelength. So light and all this electromagnetic radiation obviously behaves like a wave. You can have a short wavelength, you can have a long wavelength. Yeah, light is a wave. It's also a particle. So this is quantum mechanics, and again, this is not a physics class, so take a physics class maybe if you want to learn more about quantum mechanics, but the short version of quantum mechanics is that light is both of these things. Light has a wave nature, it behaves like a wave, it has properties of a wave, and light has a particle nature. In many ways, it behaves like a particle. When we're talking about its particle nature, these little particles of light we call photons, and we're going to see these in later figures. We're going to see little packets of light, little dots of light or whatever. These packets of energy are called photons, and yes, they carry energy with them. So yeah, light's a wave and a particle. Now, more background here. So when we talk about photosynthesis, we are going to be talking about pigments. So a pigment is defined in the key terms. Sorry for the paper rattling. A pigment is a molecule that is capable of absorbing certain wavelengths of light and reflecting others. So what this looks like is, here's a leaf. A leaf looks green to us because it absorbs the red, orange, yellow, blue, indigo, violet wavelengths of light, but it does not absorb the green wavelengths of light. It bounces them back, so you could see this with your eyes. And this is how all colors work. They absorb certain wavelengths of light and reflect others back. So we're going to see pigments involved in photosynthesis, but yeah, there are also pigments in in your shirt or whatever. If you're wearing a red shirt, it looks red because it's bouncing red to your eyes and it's absorbing everything else. But yeah, key terms definition of pigment: absorbs certain wavelengths, bounces back others. The pigments that we care about in photosynthesis are called chlorophylls and carotenoids. As always, don't memorize this exact specific structure, but it's always nice to see what these things really look like. Chlorophylls are going to harness the energy of photons. So these are the types of pigments that are going to absorb photon energy and actually use it. Chlorophylls harness the energy from photons for the process of photosynthesis. Carotenoids have a slightly different job. So these are still pigments. Carotenoids are not absorbing photons in order to harness that energy. Carotenoids are absorbing photons to prevent the rest of the cell from getting burned out. So visible light is, very useful. There's a lot of energy here, but in many ways, this is also a deadly laser. This is also very dangerous. This is an incredible amount of energy that comes in with these packets of energy, with these photons. So the chlorophylls are going to make use of this energy. The carotenoids I like the phrase, dispose of excess energy. These sort of protect everything else by dissipating any extra energy that's coming in from this light. So these are both examples of pigments, but they have slightly different jobs in the cell. Now, if you want to do photosynthesis and harness the energy of all these photons coming in, you don't want one chlorophyll molecule. You don't want two chlorophyll molecules. You want as many of these as you could possibly have. You want as many of these things as you could possibly cram into the cell to grab as many photons as you possibly can. So we're going to see hundreds of chlorophylls and carotenoids arranged into things called, appropriately, light harvesting complexes, kind of like a solar panel array, but in the cell. Here is a light harvesting complex. Each one of these little green circles is meant to be, one of these complicated molecules. But yeah, here's a light harvesting complex. Many of these individual pigments put into an array. The light harvesting complexes themselves are part of a larger structure called a photosystem. We'll talk about what's happening here in just a minute. Right now I'm just putting out the terminology. These are pigments in a light harvesting complex. These are a couple light harvesting complexes, part of a photosystem. So yeah, hundreds of these things arranged into light harvesting complexes, which themselves are part of structures called photosystems. Okay. We got a bunch of terminology out there. We are finally ready to start talking about the first half of photosynthesis, the light-dependent reactions. So here's an overview. As complicated as this looks, I always like having some sort of overview that we can go back to, some sort of big picture thing for us to look at. And yeah, there's a lot going on here. Let's focus on one part at a time. So you may notice this little thing looks familiar. We just looked at this. This is a photosystem. Oh, here's another one. There are two photosystems. So let's start with the first one. Let's begin with this photosystem here. The first photosystem in this process is called photosystem 2. Wait, no, I didn't mix up my slides here. I didn't mislabel it. The first photosystem we are going to encounter is indeed named photosystem 2. And the second one is called photosystem 1. They are named for the order in which they were discovered. So the reactions and the process of this one was just discovered first, and then this one was discovered. It's dumb. I don't name these things, but please don't let this confuse you. We are indeed starting with the one called Photosystem 2, in Roman numerals by convention. So what happens in Photosystem 2? Well, we've got photons that come down in the form of lightning bolt here. This is meant to show, light coming in. Photons are going to strike these light harvesting complexes, and hopefully this energy is going to be absorbed by one of these chlorophyll molecules as part of this complex. Now, when this photon hits this chlorophyll, that is going to cause the electrons in this chlorophyll to jump to a higher energy state. Yes, it's this concept again. I introduced this in the previous chapter. We're seeing it again here. You should, again, this is difficult to conceptualize, but electrons can exist in a low energy state, or electrons can exist in a higher energy state. So in these chlorophyll molecules in the photosystem, here we have low energy electrons, photon comes in, hits the atom, electron is excited, it is now at a higher energy state. Again, I admit this is difficult to conceptualize. That's what's happening when the photon hits the chlorophyll. So to summarize, again, I'm going to number these because there are going to be several steps here in the LDR. Photons strike pigments in photosystem 2 and excite the electrons of those chlorophylls to a higher energy state. From here, that energy, again, focusing on this pathway here, the energy of these excited electrons is going to be passed from one chlorophyll to the next, to the next, to the next, to the next, in a somewhat rambling, random fashion, until we finally get to a special chlorophyll called the reaction center chlorophyll. At the reaction center chlorophyll, something special is going to happen, but let me, write this first. The energy of excitation is going to pass from chlorophyll to chlorophyll to chlorophyll until we get to the reaction center. Very importantly, I have underlined the phrase energy of. This little arrow here is showing the transfer of energy from this chlorophyll to this chlorophyll. The electrons, in these steps, in these passages, the electrons are not physically moving, or they're not being transferred from one molecule to the next. It is simply the energy on these electrons that's being passed from this chlorophyll to this chlorophyll. So this chlorophyll has high energy electrons. It's going to pass its energy to this next chlorophyll. So this first chlorophyll is going to power down, go back to low energy electrons. And now this chlorophyll, which has received that energy, is going to power up to high energy electrons. So again, it's a small little phrase here, but it's important to note that along here, the electrons are not being physically transferred. It's just the energy that's being passed from one chlorophyll to the next, to the next, to the next, until we get to this special reaction center, or RC chlorophyll. So energy of excited electrons passed from one chlorophyll to the next until it gets to the special reaction center. At the reaction center, something different happens. At the reaction center, chlorophylls are now going to be physically passed. Or I'm sorry, did I say chlorophylls passed? Electrons are going to be passed physically at the reaction center chlorophyll. So now, again, doesn't happen with any of the other chlorophylls. At the RC, or reaction center chlorophyll, it is going to send its high energy electrons through something called an electron transport chain. Yes, it's this thing again. So again, calling back to the previous chapter, it is that same sort of chain. The high energy electrons on the reaction center chlorophyll are going to be passed to a carrier, to another carrier, to another carrier, to another carrier, to another carrier. And we'll see that in just a second. But before we follow this to its final destination, we have to think about this reaction center chlorophyll. It lost its electron. It physically sent its high energy electron through these series of carriers. It is missing its electron. If it wants to receive excitation energy again, it's got to replenish its electron. So the reaction center chlorophyll gets its electron back from water. It breaks H2O apart into O2, some loose protons, some hydrogens, and electrons, which it can, again, replace its physically lost high energy electron. So it seems like kind of a side note, but a very important one about what's going on here. This is why H2O is required for photosynthesis. This is why O2 gas is generated as a byproduct. It's to get its electrons back on this photosystem 2's reaction center chlorophyll. Let me summarize this. The RC chlorophyll delivers its high energy electron through an electron transport chain. The RC chlorophyll replaces its lost electron from water, H2O. H2O goes to O2 plus electrons. Those electrons go to the reaction center chlorophyll. Okay, so now on to this thing. So yeah, it's the same sort of concept here. A lot of this I just copied and pasted from the previous chapter. The high energy electrons are passed through a series of carriers. They are lower energy with each passing. This energy is used to pump protons across a membrane, create a proton gradient. That proton gradient is going to power ATP synthase. Again, all these same slides from the previous chapter. Hopefully, this is just review at this point. So here's that summary. Energy used to pump protons, creates a proton gradient, used by ATP synthase to generate ATP. And again, we see this in our big overview. Yeah, there's photosystem 2, there's the electron transport chain, there are the protons being pumped from one side of the membrane to the other, and there's ATP synthase utilizing that proton gradient to generate ATP. So again, I'm breezing through this because we just talked about electron transport chain and ATP synthase in the previous chapter. Okay, so in the previous chapter, the end of this electron transport chain was, being sent to oxygen gas to form water. It is a little different here. Instead of going to oxygen gas to form water, in here, the end of this electron transport chain is photosystem 1. So remember, two photosystems. We're on the second one here, photosystem 1. So here's photosystem 1 receiving low energy electrons from this electron transport chain. Low energy electrons arrive at photosystem 1's reaction center chlorophyll. What happens in photosystem 1 now? Well, exactly the same thing that happened in photosystem 2. Photons are going to randomly strike chlorophylls in these light harvesting complexes. They're going to pass their energy from one chlorophyll to the next, to the next, to the next, to the next, until it makes it to the reaction center chlorophyll. And now you have high energy electrons in the reaction center chlorophyll. So, in summary, photons excite the electrons in photosystem 1's chlorophylls. This energy moves to photosystem 1's reaction center chlorophyll. Here's where it's a little different from photosystem 2. Once these electrons on the reaction center have been energized, here in photosystem 1, they're not going to be passed through an electron transport chain. Here in photosystem 1, these high energy electrons are going to be passed on to NADP plus to form NADPH. It's another one of these. It's another electron carrier. This is nicotinamide adenine dinucleotide phosphate. Oh, there's a little phosphate group down here. It's just like NAD and NADH from the previous chapter, but there's a phosphate group down here. It's a slightly different molecule. The P does not stand for photosynthesis. but if it helps you to not get them mixed up, maybe you could pretend that it does, because NADP plus and NADPH are involved in photosynthesis here. So, yep, we're taking these high energy electrons from photosystem 1, putting them on NADP plus to form NADPH. We now have a high energy electron carrier, which we could use in later steps. But in summary, the high energy electrons in photosystem 1 are used to reduce, there's that term again, going to use these throughout these couple chapters, reduce NADP plus to NADPH, which is just yet another one of these electron carriers. So again, this seemed intimidating at first, but hopefully you can understand this overview here. Photosystem 2, electrons come in, photons come in, excite electrons, energy is passed to the reaction center, the electrons are physically passed now from the reaction center chlorophyll through the electron transport chain, pumping protons, powering ATP synthase. The low energy electrons now arrive at photosystem 1, they're excited again, and they're passed on to NADP plus to form NADPH. I like this overview figure. It shows the membrane, it shows the protons, it shows all of this together. I also like this summary. It's just from a different textbook. I like this summary because it shows the energy ups and downs of all of this. So here's photosystem 2 receiving energy from a photon, and yeah, this is showing the electrons going from low energy to high energy. Now they pass through the electron transport chain, they're back down to low energy again, and they get excited a second time. Now they're at high energy, and they're passed on to NADPH, where they're high energy electrons on this valuable high energy electron carrier. So I think both of these are good visuals for understanding what's going on here. And if we want a halftime report, I know I did an overview of photosynthesis as a whole in the beginning of this. If we want a report of what we've done so far, here is what has happened in the light-dependent reactions. In the light-dependent reactions, we took water and photons, light energy, and we ended up with oxygen gas. Again, that's what happens when we ripped electrons off of H2O. We've ended up with some oxygen gas, some ATP, which we generated down here from ATP synthase, and some NADP+. Notice, we haven't messed with carbon at all. We haven't built anything. We haven't, constructed glucose. The whole purpose of this LDR is to get these valuable molecules. We're going to use ATP in the next stage. We're going to use these high energy electrons in the next stage, but the LDR is just about harnessing energy from photons in order to get energy in these molecules, which we could use in the next stage. So here's our overview so far. All right, now that brings us to, of course, the other half, the LIR, the light-independent reactions. Now, before we get too deep into the light-independent reactions, I do want to point out real quick where this is happening. So the light-independent reactions are going to take place in what's called the stroma. The stroma is essentially the cytoplasm of the chloroplast. Labeled here, aqueous fluid. It's just the water inside the chloroplast. This is in contrast to the light-dependent reactions, which were embedded in the membrane itself. Here, the reactions we're going to see in the LIR are just not associated with a membrane. They're floating around in this stroma. So yeah, LIR in the stroma of the chloroplast. I also want to note about the LIR, I'm going to number the steps here, just to count through this. I do want to note the first 3 steps of the light-independent reactions are called the Calvin cycle, named after Melvin Calvin, the scientist who first described this. Yeah, Calvin cycle just refers to the first 3 steps of the light-independent reactions. So what is the Calvin cycle? Well, it's another one of these. It's another one of these big old metabolic pathways. There's a bunch of enzymes, a bunch of intermediates here. We're not going to memorize all of these things, but we do need to know, just like you did in the last chapter, the first step. And this first step up here is one of the most important chemical reactions in all of life on the planet. This is a chemical reaction that is going to take inorganic carbon. Remember, CO2 is an inorganic molecule. It's going to take this inorganic carbon and it's going to make organic carbon. No other chemical reaction does this. No other enzyme does this. No other pathway does this. This is really important. In fact, there's a name for this process of going from inorganic to organic. It is called carbon fixation. So this enzyme here at the very beginning of the LIR, it's got a funky name, and there's a reason for it. This enzyme is called Rubisco. You don't have to capitalize it like this, but there's a reason for it. This enzyme called Rubisco performs carbon fixation. And carbon fixation, if I can read from the key terms, the process of converting inorganic CO2 gas into organic compounds. So it attaches CO2 to something called ribulose bisphosphate. And I know there's a bunch of chemical names here. I'm not a fan of making you memorize too many of these. Just remember, there are a lot of these. I'm only making you know a few of them. So there's CO2. There is the 1, 2, 3, 4, 5 ribulose bisphosphate. 5 plus 1 equals 6, but actually that 6 immediately falls apart into 3 plus 3. So CO2 attached to ribulose bisphosphate, creating 3-phosphoglyceric acid, or 3-PGA, X2. So if you're following the math here, this is 5 plus 1 becomes 3 plus 3. CO2, ribulose bisphosphate, becomes 2 copies of 3-PGA, or 3-phosphoglyceric acid. This is just the first step. This is just what this enzyme Rubisco does. And yeah, if you want to know why it's got the funky capitalization, it is ribulose, R-U, bisphosphate, bis, CO2, co. It's the Melvin Calvin thought he was being very clever here, but yeah, it's named after ribulose bisphosphate and CO2, the enzyme Rubisco. Okay, so what happens next? Now we've got this phosphoglyceric acid, and here is where we spend that ATP and we spend that valuable NADPH. Both of these things came from the light-dependent reactions. We're going to spend that creating glyceraldehyde 3-phosphate, G3P. So here we go. Oh, sorry, I forgot I did this. Ribulose bisphosphate CO2. Very clever. ATP, so step number 2. ATP and NADPH, again from the LDR, are used to reduce this 3-phosphoglyceric acid to glyceraldehyde 3-phosphate. We haven't added or removed any carbons. We're going from this 3-carbon molecule to another 3-carbon molecule. Now, some of these G3Ps are going to go off to form glucose, and we will follow that journey in just a minute. But this is supposed to be a cycle. We're supposed to end up right where we started again. So we somehow need to get back to this 5-carbon ribulose bisphosphate. The third and final portion of this Calvin cycle is getting back to that again. We're going to use some of those G3P. So yeah, we're doing this many, many, many times. We're creating lots and lots of glyceraldehyde 3-phosphate. Only some of it goes on to form glucose. Most of it goes towards regenerating ribulose bisphosphate again. So we're going to take some of these G3Ps and use some more ATP, again from the light-dependent reactions, to regenerate ribulose bisphosphate so Rubisco can do its carbon fixation thing and we can continue on with this cycle again. So step number 3, ATP used to regenerate ribulose bisphosphate from some of that glyceraldehyde 3-phosphate. So again, these first 3 steps are the Calvin cycle. The Calvin cycle generates G3P, but we're not done yet. We have to use this G3P to build stuff. So this is not the Calvin cycle. These 3 steps were the Calvin cycle. Our 4th step is outside of that. And here is actually where we move into the cytoplasm. I stated near the beginning of this chapter that virtually all these steps were going to be in the chloroplasts. This is the one that is not. This very final step is just in the cytoplasm of the cell, where some of this glyceraldehyde 3-phosphate is used to build all this stuff. Again, it's a pretty useful 3-carbon molecule here. You could build amino acids with it, you could build lipids with it, you could build sugars with it, but we are supposed to get to glucose. That was the point of this. This is our end goal. We need to figure out how glyceraldehyde 3-phosphate specifically becomes glucose. Well, it is going to be converted to glucose through a metabolic pathway called glycolysis, which should sound familiar to you and should be confusing to you. Because we talked about glycolysis in the last chapter, and this was a breakdown pathway. This is, going from glucose down to pyruvate. Why is this involved now in building glucose? Well, this is not true for every enzyme. Some enzymes, under the right conditions, can be shifted, just like a car, can be shifted into reverse and do the opposite reaction. They can go both ways depending on the circumstances. And as it turns out, these early enzymes in glycolysis, again, we first talked about this in the last chapter as breaking down glucose. These first few enzymes can be shifted into reverse to build glucose. And we didn't memorize these intermediates in the last chapter, but if you look closely at this intermediate here, wouldn't you know it, it's glyceraldehyde 3-phosphate. So we could take a couple of these, shift this enzyme in reverse, shift this enzyme in reverse, shift this enzyme and this enzyme and this enzyme in reverse, and we can make glucose from glyceraldehyde 3-phosphate using this same familiar metabolic pathway, just shifted in reverse. So these are the same enzymes from cellular respiration, just working in reverse. And we finished, right? The whole point of this was to get to glucose, and we got there. So if we want an overview just of the light-independent reactions, we took CO2, again, our building block, our inorganic carbon. Rubisco made use of this as a substrate. We used up ATP, we used up NADPH, and we created glucose from this. So we're done. A couple of overviews, though. Again, I like a big overview slide just to kind of summarize everything. I like this one because it's showing light-dependent reactions over here. Yep, these are the stacked membrane discs in these chloroplasts, and this is showing generating ATP and NADPH. It's also showing water going in and O2 gas coming out. So yeah, very convenient overview. And then over here, we have the light-independent reactions, which includes the Calvin cycle, taking the ATP and NADPH from the LDR, having an input of CO2, and glyceraldehyde 3-phosphate as the end product of the Calvin cycle, which can then go off to form glucose through glycolysis. Here is another overview. Again, I'm not trying to bog you down with a bunch of information, but I hope that at least one of these is just a good, convenient summary of everything. It's always good, to, I think, to have a big picture of what's going on here. And yeah, this is showing the same thing, but just in a slightly different way. Light and water coming into these light reactions, generating O2 as a byproduct, generating ATP, generating NADPH. These things feed in to the Calvin cycle, which takes an input of CO2 and this energy and builds glyceraldehyde 3-phosphate, which here in the cytoplasm can be used to build glucose. So nothing to write down from these. These are just, I think, good summaries. But yeah, that takes us to the end of photosynthesis and to the end of recorded lecture 2-4.